actually work at the scientific computing program, which is a branch of your group which deals with the application of application and development of statistical methods to biological problems. So these are my advisors. So Carvalho is a PhD from Harvard in population dynamics, and Leo is a very good statistician in Sheffield and West Asia. So Alexander already did a very nice introduction. So I'm actually a microbiologist, a very special kind of biologist. And I worked as statistics assistant at the Pan American Health Organization for 3 years. And lately, I've been working at the scientific computing program, and I was approved to be a master's student in the statistics in my university. But unfortunately, fortunately, I won't be able to complete my degree. I'll take some coursework, but I'll be moving to the University of Edinburgh to pursue a PhD in evolutionary biology. Usually I don't do this type of introduction, but since none of you know me, I thought it would be nice to introduce myself. So nice to meet you. So I'll move on to the motivation of this work, why we're doing this. And you know, the deterministic models, and here I really mean differential equation-based models, are widely used in science, engineering and all, but let alone in biology. Every aspect of biology has been modeled, has been subject to modeling by deterministic approaches. So population growth, disease spreading, these are the most interesting in my opinion, but you can also use these models to model cellular and molecular interactions. So the thing with these kind of models is that they provide a crude but easily interpretable representation of reality. So they are usually very silly in their representation, but they have also the desirable property that you can directly interpret their parameters. So moving on to another important aspect, we have that temperature is obviously a key factor to the growth of several organisms. So disease-carrying bugs, which are important for many diseases like Chagas disease and dengue, and yellow fevers and so on. Pathogenic bacteria, which grow in food and stuff like that. And of course, economically important plants. So with the deterministic model and some time series, how can you learn about model parameters? So what I'm going to do here is that I'm going to offer you a Bayesian approach to this problem. It's nothing really groundbreaking or new, but I really find it worth showing. So let's see some background here. So consider you have a deterministic model of very generic form, and you have a set of inputs that pertain in the real line, in the real hypercube, and a set of model outputs. So I purposely specified P different from N here, so that we can be very general about our formulation. So the model connects X and Y through a structure, of course, in a set of parameters theta, which lives in an ordered hypercube in real space. So these formulations, this notation is originally from Poole and Raftery in 2000. To the best of my knowledge, this is the first attempt to do Bayesian learning on these models. So in our particular case, we have laid our dirty hands and some data Y and some inputs X that we think can be modeled in this way. So of course we're interested in learning about theta. I'm sorry. So this is an idea for this problem. So we're interested in population growth. So this is a very classical differential equation. Those of you who have taken courses in differential equations most probably have seen this equation. So it's the Verhoeven's equation, and it models logistic growth of population. So this is the limiting factor. This is the carrying capacity, so it's the maximum the population can achieve, and this is the growth rate. So fortunately we have an analytical solution to this equation, which is this one. And what I did is I naively assumed, so this is very important, I naively assumed that temperature does not, in our setting, depend on time. This, I know, is a very realistic assumption, but for it to work mathematically, you have to assume that. So what I did is that I formulated both K and R as functions of temperature, so that we could model temperature-dependent growth. So to complete my model specification, what I did is that I assumed one very flexible form for dependence of these parameters on temperature, and it's a Gaussian kernel. So what we have here is that we will have a maximum point to minimum point, because that's how it usually works. At low temperatures, it's not very nice to live, and at very high temperatures, it's not nice as well. So there is a middle ground where population growth is optimal. But by allowing different parameters, I can make these parameters to have different maximums, for example, on different variabilities. So these are my set of parameters where I'm interested at. So here is another controversial assumption. I assume P of t, so the population over time temperature, to be a Gaussian process with fixed variance. You can criticize me for that, and I will accept it gladly. So the point is, we have a set of observations, and we have also the times, precisely, the times and the temperatures where those observations were measured. So this monster here is actually my likelihood function. I mean, my sampling model. So my population size will depend on temperature, time, the initial condition, and my set of parameters. So this is more or less like what is going on here. So I'd like to stress that the normal formulation makes it possible for me to write my model in this form. Okay? So we have a deterministic part and a stochastic part. So after giving you this monstrous likelihood function, to make it up to you, I came up with some nice priors. Some biologically motivated proper priors. So they were listed, I'm not really a specialist, but I worked with specialists, and they were listed to reflect the biological behavior of those functions, but remain diffuse, so we could actually learn from the data. That's, I think, an important aspect. So, of course, we have normal priors for the location parameters, so to speak, so to make an analogy with Gaussian, Gaussian distribution. And these parameters, of course, has to be positive. And I'd like to call your attention here, because this, of course, has to be positive. You cannot have negative population. And but this can be either positive or negative, because your population can either increase or decrease over time. So this is, or the monster here is my posterior. And I don't want you to pay attention to the expression, I just want you to realize that the model for the population is hierarchical because it depends on two unobserved quantities. I don't really observe these two guys over here. So this is another nice aspect, I think, which is how we actually did posterior approximation. How we did approximating that posterior. So we use something called hybrid or Hamiltonian Monte Carlo. And I won't explain in any detail what it is, but the point, the desirable properties is that it avoids random walk behavior since there's no random walk step in this algorithm. And it allows, as a consequence, it allows big moves with high acceptance probability. So you can actually move far away from your actual proposal and still have a decent acceptance probability. And another type of probability is that it does not suffer from a highly correlated posterior. So your posterior can be highly correlated and it will converge in reasonable time. So we used this package, Stan, which was named after Stanislav Ulam, which was a great mathematician and father of multicolor methods. So in R, and the neat thing about this package is that it's implemented in C++, and it uses a very nice sampler, which I'll discuss more of its properties afterwards. And it has a neat syntax that allows you to easily port your code from bugs to its syntax. And it has a very nice interface with R. So our MCMC was run for 50,000 iterations, discarding about half as burning, and we assessed convergence by traditional methods, so it's really important. So the first results I'd like to show you are the simulation study results. So for a set of parameters that I chose, you create a grid of temperatures and times and temperatures, I'm sorry, and generate a big number of data sets of this size. So of course the size of the data set is the size of the grid, by sampling from this model, which we can of course do. So after that, you run your algorithm for all the data sets, and then you come up with this table. So here we have the true value that I specified. Here we have the posterior mean we obtained, and here we have some frequencies per bus, so the bias and the mean square error, and the coverage of our credibility intervals, which I think are quite good, except for a few. I think it's decent. It's reassuring. So we have this a real application, real-world application, which is for Rodius prolixus. So Rodius prolixus is one of the vectors of Chagas disease. It's a very important disease-carrying bug, and one of our fears is that it's gonna colonize other areas with climate change. That's the main motivation. So in order to catch a glimpse, to get some insight on the population dynamics of rodents with respect to temperature, is that we measured the population sizes in 10 temperatures in this range, this big range, for 35 days, and we started with 20 females for all temperatures. So that was performed in Microbiology Institute in the university. So this result here is the posterior and the prior distribution for these quantities, for the carrying capacity and for the growth rate. So I think it's clear. I think it's clear for everybody that we actually learned something about the data. About the process from the data and in relation to our prior expectations. Here we have the posterior mean and associated credibility intervals, and the prior mean for all parameters, included variance. So what we can see is that we estimated a very big variance, but for example, for this parameter here, our prior expectation lied within 20, but we actually learned that it's much higher. That means carrying capacity is much more variable over temperature than we anticipated. So that's a nice result, I think. I mean, one thing I want to stress is that all these guys have a nice interpretation, physical interpretation. That's, I think it's a neat result. So this is the posterior. So here we have temperature, and here we have time. So this is the data, and this is the posterior. So our posterior agrees to some extent to our data. The figure's not really good, but I think in the paper is better. So let's jump to some conclusions, and let me wrap up a doggy bag for you to take home. So, of course, we stress the importance of using Bayesian inference to learn about model parameters. I'm not trying to make politics here, but I think that the Bayesian way is the best and more elegant way to learn about these, to study this problem. Because we can actually incorporate expert opinions on these parameters and then learn about them from data. I think it's the most elegant way to do it. And of course, parameters retain direct compatibility. I've stressed this over and over. HMC turned out to be a very efficient algorithm. And the nuts, so the no U-turn sampler, it actually drops one of the major weaknesses of HMC, which is hand-tuning of some parameters. So the nuts algorithm actually drops that drawback, so it doesn't have it. And we have observed, although not presented, a considerable speed up and quicker convergence using this algorithm. So some perspectives here are to make the variance of the process dynamic, so it will change over time, some smooth function over time. I think it's going to improve model fit, and it's also more realistic. To apply this model to other data sets, for example, bacterial growth, I think we have a nice application for this in studying bacterial growth. And another perspective is to do a complete treatment of acidity using a technique called Bayesian modeling, which, in a nutshell, what it does is that you actually model the uncertainty you have about some parameter, and the uncertainty this model structure induces in your parameter, in your quantity of interest. So you actually meld the information of these two prior or posterior distributions. So thank you, those are the references. I would like to thank my advisors, also my friend Leonardo from the National Space Research Institute, and the staff of the computing program who provided me an office with conditioned air and a computer terminal and a very nice gourmet coffee machine. Thank you very much.